about modeling nanocargo in solid tumors, the core shell approach. So here is my agenda for this talk. I'd like to cover all the topics today. And let me start sharing with you what I found in a literature search about the number of papers published in vivo bioassay. So I did this search at the beginning of this year, January, and also I did it again last weekend. So here you can see the numbers. So in January, we have a little bit over 1.5 million papers published in vivo bioassay. And 3 days ago, I found an increase from a little bit over, perhaps about close to 70,000 papers added. But what is interesting in this data is that from all the papers published in this topic, so we found that only in January, only about 3% of the papers included some kind of mathematical modeling of the data. So, and then 3 days ago, we found a very good news. It improved a little bit from 3% to 4%, which means the research community working in this area shows a trend that I pick up on the plot on the right-hand side, lower part of my slide. So you can see an increasing of the numbers of papers that included any mathematical model. So this plot was the plot from January. And so it is indeed a very good news. So we are increasing the number of papers and the authors that use any kind of mathematical model to explain the experimental data. This is a very challenging aspect of this kind of research, because we are trying to move from a simple qualitative analysis to a more sophisticated quantitative analysis. So, and this means a very good news in this regard. So let me show you quickly what kind of data you can see in the literature. This is just one paper published this year. And this is a plot of this paper that shows this is an experiment that they use mice to capture nanomaterials in the air. So, and how this nanomaterial goes into the animal's body. In particular, they use PET scan data to check the zirconium-89 isotope, it's a PET isotope. And they found, as you can see in this plot, they found that most of the particles, they go to the bone tissue. And the blue data you can see here, this is typical. So the authors just say, okay, so the time required for the concentration of the nanoparticles in the mice's bone, it takes about, let's say, 45 hours to reach the maximum. However, there is no precision in describing this data.
Não tem problema, calma. Você entra de novo. Tá aqui, ó.
Yeah. Is there any question, please? Okay. So this is very standard in the literature. And I can tell you, we modeled a similar data like this one. So you can see the concentration increasing, reaching a maximum point, and then decreased afterwards. So we modeled this kind of data about 20 years ago. And still this day, people are using only, sort of qualitative analysis of the data. So it's it was a very good news looking at the previous slide. And we could see that the number of papers using mathematical model are increasing, and it is increasing as a function of time too. So it jumps about 1% in less than 1 year. So let me show you one data, very simple data. This was published about more than 20 years ago in the Biophysical Journal. So this is one experiment we did. So we injected in the animal's tail magnetic nanoparticles, and we were able to monitor the concentration of the nanoparticles in the blood circulation. And we observed the concentration of nanoparticles decreasing in the blood circulation as a function of time. So you can describe this data very easily. If you take a plot like this, a semi-log plot, the concentration versus time here in minutes, so you can see the open circles are the experimental data. So we collect the blood from the animal and every 5 minutes, something like this. And we found the half-life time of the disposition of nanoparticles in the blood circulation is about 7 minutes. So we have a very precise data saying about the half-life time. And to describe this experiment, we can use a very simple mathematical model, which means the rate of decreasing of the concentration of the nanoparticles in the blood circulation. It goes linearly with the concentration of the particles in the blood circulation. So this is a very simple differential equation you need to solve to find that the concentration of nanoparticles in the blood circulation goes exponentially with time. So, and in the plot, you can see the black solid line is exactly the best fitting of the data. And the experimental data are the open circles. You can see the best curve fitting. And then you can extract the half-life time, which is about 0.7 over K naught. K naught is the constant you extract from this fitting. So this is a very simple model, very simple. We can sophisticate a little bit more the experiment. So instead of looking only at the nanoparticle concentration in the blood circulation as a function of time, we would like also to investigate where those nanoparticles go after they leave the blood circulation. Actually, they go to different biological compartments. They go to one compartment number one, compartment number two. It could be the brain, could be the lung, kidney, spleen, stomach. So you can track the concentration of nanoparticles going, leaving the blood circulation and moving to different biological compartments. At the end, you can model this problem using one differential equation for each compartment. In this scenario, actually, we have two different but complementary mathematical model. One is the model we call in parallel. So what that means, this scheme can show what the parallel model means. It means the blood circulation transferring material to one compartment, to the second compartment, to the third compartment, and so on. So the transferring from the blood circulation to different biological compartments, it takes place in parallel. So you can, again, solve the differential equation for each compartment. And you find, actually, this is the concentration at one particular compartment we call M. It could be lung, it could be the brain of the animal, it could be the spleen or the liver of the animal. So the concentration goes with an expression like this. But I'd like to emphasize the important parameters here, which are the typical times. So you have the loading time, which tells you how fast the material or nanomaterial goes out from the blood to a particular compartment. This is the loading time of that particular compartment. So this is a very important parameter you can extract from doing your fitting, the fitting of experimental data using this equation. The second parameter, which is very interesting too, this is the loading time. I'm sorry. T naught is the loading time, is a typical time transferring from the blood circulation to one particular biological compartment. So besides the loading time, you have the transient time. So TIM is transient time, like this one here, TI1. The transient time is the typical time the material stays inside the biological compartment before being disposed from that particular compartment. And the time, the disposition time, is the third very important parameter you can extract from this math model and in the scenario of the parallel transferring. So we are transferring materials from the blood circulation to different organs, and the scenario, in this scenario of parallel. So besides the parallel modeling, we can also analyze the system if the transferring of material goes in series. So here is, you have the loading time, which is the typical time of transferring from the blood circulation to one compartment, the first stage of compartment. And then from the first stage, it goes to the second stage. So it goes in series from the blood circulation to the first compartment, the second, and the end compartment. So you can track this, you can solve the differential equations for each compartment. And if you do this solution for two compartments, those are the two equations you can extract from this problem. So again, you have typical, the most important parameters are the typical times. And T naught is the loading time, is the typical time from transferring material from the blood to the first compartment. And then you have, in a sequence, you have the transient time from one compartment to the next one. And finally, you have the disposition time for between the last compartment and to the extraction of the system of the animal, which goes to the feces or the urine of the animal. So at the end, you can find a way to track and measure the concentration of your material in each biological compartment. And then you can fit the data as a function of time using a set of differential equations. And then you extract the important parameters, which are the typical times. So in this regard, I want to show you a real experiment. So we did an experiment. We analyzed a system like a combination of one compartment in series and coupled to this one compartment in series, we have two compartments in parallel. Actually, we injected one material in the animal circulation, blood circulation, and then this animal has a tumor, and then we analyzed transferring of this material to the tumor, and actually we had to model the tumor as two compartments. So the tumor is a very complex biological system, but to analyze the data, you can make this first approach. So the tumor is split in two compartments, the shell around a core of the tumor. So the two compartments are the shell and the core. So in such a way that the nanomaterial or your material comes to the tumor first in the shell, and then it is transferred to the core of the tumor. So these shell and core, they work in series. At the same time, we want to track the destination, the fate of the material in two extra organs. We choose the lung and the liver to analyze our data. So if you look at what happens with the material that comes to the shell, it is described by this solution of this differential equation, and then it is transferred to the core of the tumor, this is the differential equation regarding to the core, and this is its solution. And for the two other compartments like lung and liver, here are the differential equation we need to solve. So what was the design of this experiment? Actually, we worked with two kinds of solid tumors, the earlish and the tumor provided by 4T1 cell line. So we did a intraperitoneal injection of one infrared dye, so the free molecule of the infrared dye. And at the same time, we made a formulation of this infrared dye using a nanoemulsion. So in other words, we use two materials to inject in the animal. And then we analyze animals bearing different tumors. So, and then we were able to find the concentration of the material in the tumor shell and also in the tumor core. At the same time, we analyzed the concentration of the materials, both the free dye molecule and the nanoemulsion of this free dye in the liver and the lung for both kind of tumors. So what were the experimental data we found? For the earlish tumor, and we found in the tumor, the concentration of the nanoemulsion, which is the red data here, the concentration of the nanoemulsion decays as a function of time in the earlish tumor. The same trend we found while analyzing the 4T1 tumor. So the nanoemulsion concentration decays as a function of time. On the other hand, the free molecule, which means the free dye, it slightly increases as a function of time in both kinds of tumor. actually the data that I show in the upper plots here are the data collected from the whole tumor, but also we analyze the data recording the concentration in the shell part of the tumor, also in the core part of the tumor. And then, of course, each of these data can split in the shell and the core. When you split the data, you see for the earliest tumor, we see the core, the tumor shell going downwards and the tumor core going slightly upway. And we check this in the nanoemulsion and also in the free drug. If you look at the free drug data, it goes slightly increasing as a function of time for the free molecule. So it's quite different from the nanoemulsion formulation. And we see the same trend splitting the tumor in core and shell. So it is important to stress here that symbols are experimental data, and the solid lines, red and blue, are the best fitting using the equations for a series transfer from different compartments here. So different compartments here means the shell and the core of the tumor. Of course, we did the same analysis for the two organs we choose, which is the lung and the liver. So here on the left-hand side are the data for the nanoemulsion, the red dots, and for the free drug. So these are the data. The up here, left-hand side upper plot is for the liver, and the lower plot is for the lung. This left-hand side is for the earlish, and the right-hand side is for the 4T1 tumor. So again, it is important to emphasize that the solid lines are the best fitting for equations in the parallel model. So here, you can extract from all these fittings, you can extract the typical time. So the loading time, which is the time, typical time for transferring material from the bloodstream to the biological compartment. We have the transient time, which is the time, the typical time the material can stay inside the biological compartment, and the disposition time, which is the time, typical time, the material can be extracted from the biological compartment. So, of course, from this typical time, you can extract the half-life time for any kind of time you are looking at. So, in this regard, we can just emphasize some very interesting conclusions. And again, you can extract data with much more confidence than if you are not using a mathematical modeling. Okay, so you can see a huge difference between the disposition time from lungs and the disposition time from liver. So you can see typically in the case of liver, comparing liver and lung, you can see that the disposition time from the liver, if you look at nanoemulsion in the liver, is about 1.9 hours, and in the lung is about 3 hours. So it's almost 50% longer, the time of disposition from lung. If you compare the same tumor, which is earlish tumor, from liver and lung. So time disposition is very different. So you never can get this data numbers if you are not using a mathematical model. At the same time, so you can see these not only for the nano-emission formulation, but also from not only for Ehrlich, but also from the 4T1 tumor. So again, the disposition time from liver and lung. So lung is roughly 50% lung. So at the same time, you can also compare the typical disposition time when you compare the free drug or the free infrared dye with the nano emulsion. Is quite different. So is about half the disposition time for if you compare Ehrlich with the 4T1 tumor. So you extract very, very important data from your feeding. So if you look in terms of the tumor, if you can compare the typical time, and here, which is the transferring time from one compartment to the other one, which means transferring from the tumor shell to the tumor core. So you can see a huge difference, and transferring from the tumor shell to the tumor core. So this is a typical transient time, and you can compare the 4T1 tumor with the Ehrlich tumor. So you see here about 3 orders of magnitude longer. So the nanoemulsion stays in the tumor shell much, much longer in the Ehrlich tumor. So it's about more than 1,000 times longer in the Ehrlich. So you never can extract this conclusion if you don't perform a mathematical analysis. The same thing you can see. So the typical disposition time from, if you compare the 4T1 with Ehrlich, is also about 3 orders of magnitude longer in the Ehrlich tumor. It means that these tumors, they behave completely different in terms of transferring time from the shell to the core, and also from disposition time. So all these very important conclusions you can extract from the typical data your math analysis can provide you. So here are some illustrative conclusions about the benefits of using a mathematical model to understand and analyze the in vivo data. So first, quantitative analysis does provide extraction of typical disposition time, which is very important. If you are interested to move the results of one experiment like this in terms of a future protocol. So qualitative analysis doesn't provide to you